Hey there. Previously, you guys have learned about how a muscle cell is stimulated to contract and how the sarcomeres of these contractile units of the muscle cells shorten and ultimately produce the contraction of the muscle cell. And if we zoom out and all of this is happening at the same time, the entire muscle can shorten. Okay, so very brief review here. A muscle cell, a myofiber or myocyte, is stimulated to contract by an action potential traveling down a motor neuron, triggering the release of acetylcholine into the synaptic cleft of the neuromuscular junction. Acetylcholine binds to receptors on the motor end plate, and if this depolarization is strong enough to achieve threshold, voltage-gated sodium channels will open wide up. Sodium floods in, and ultimately, we know the story, and action potential is generated in both directions along the muscle cell as well as into those T tubules. This depolarization leads to the release of calcium from its stores within the sarcoplasmic reticulum, and calcium is going to bind to troponin on the actin fibers. Right? So this right here is a sarcomere, Z disc, red is actin, Z disc, red is actin, so Z disc to Z disc, this is a sarcomere. The blue, this is myosin, we can see the little myosin heads, and the zone of overlap here. So where myosin heads can interact with actin as long as the active sites on actin are wide open. And so that's where the calcium comes in. Remember that calcium binds to troponin, which moves tropomyosin out of the way. So here, calcium bound to troponin, tropomyosin is moved out of the way, leaving the active sites wide open. As long as the active sites are open, myosin, with these little myosin heads, is going to bind to the active sites here, forming a cross bridge. And as we know, the cross bridges, let's go back up here, essentially pull this actin closer to the M line. So pulling the Z disc closer to the M line and this one closer to the M line, and finally shortening the entire sarcomere. Throughout this video, I am going to use my hands to kind of represent this process. What we have are the actin and myosin, right? So the myofilaments, which are overlapping one another. My thumbs here, these are the Z discs. And so as the sarcomere shortens, again, because calcium has bound to troponin, now the active site is open and cross bridges can form. Now, all this is happening. Now, the sarcomere can shorten. So the distance between my thumbs is now shorter than it was before. Right? So it starts out pretty far away. And because of the action of the cross bridges, the sarcomere shortens. And so when this is happening, one after the other, so thousands of these all in line, the entire muscle fiber or the cell, myocyte, myocell, is going to contract. The entire thing is going to shorten. Okay, so this is called a muscle twitch. Okay, and essentially this is a tie over from when muscle physiology was first really being studied. Essentially, scientists would take a frog calf muscle and they would stimulate it with electricity and it would twitch, right? So in that fraction of a second, that twitch, all of this stuff was happening. And so that term is kind of tied over from those days. Okay, so again, when the muscle cell is stimulated, all of these sarcomeres shorten because of these cross bridges. Okay? Keep in mind that the actual depolarization of the cell, so the stimulus itself and action potential, is actually happening a little bit before the muscle starts to shorten. And so if we look at this graph here, we can see the membrane potential. So what is the charge of the cell? Depolarization, calcium being released, repolarization, the cell returning back to resting membrane potential, only lasts a few milliseconds, just like we would see in a motor neuron. But that depolarization is only triggering the release of calcium, right? And then this has to happen, and then this has to happen, and then finally the Z discs are finally pulled together and the whole muscle cell is shortened. And so this graph up here is showing us the tension, or essentially the force that is generated by that muscle due to the shortening of the sarcomeres. And so after all of these different steps happen, this is called the latent period. Then and only then the sarcomeres shorten, generating more and more and more tension, more and more force. Think of your muscle shortening as you lift the barbell or the dumbbell. So that is happening, getting shorter and shorter. As we know, acetylcholinesterase is kind of, acting like a little Roomba, cleaning up all of this extra acetylcholine within the neuromuscular junction, recycling as much as possible. And so without the stimulus, calcium is going to be returned back to the sarcoplasmic reticulum, cross bridges are going to break, and finally those the sarcomere is going to lengthen, Z discs getting farther and farther apart. And so as the sarcomeres lengthen, the cell, of course, is relaxing, and as we can see, the tension, the force that's generated by that muscle is reducing. Okay, couple points here. The latent period is when the electrical stimulus happens. The tension, right, usually increases as the cross-bridge cycles are happening over and over and over again, and then without another stimulus from the motor neuron up here, the muscle is going to relax. Okay, so down here we can see that the muscle tension is produced because of the shortening of the sarcomeres. Now, as a whole, a muscle cell is either stimulated, it's depolarized, and so calcium is dumping out into the cell and cross bridges are starting to form, or it is at rest, and so everything is kind of going back to normal. So you would think that as far as a single muscle cell is concerned, you either are contracted and produce a certain amount of tension, or you're relaxed and you produce no tension. However, we know that with the same contraction, same exact processes happening in your muscle cells, you can lift a piece of paper, which is super light. And so that is a shortening of the sarcomeres to be able to lift your arm and lift the paper. Or you can pick up something a little bit heavier, right? Same exact motion, same processes happening in your muscle cells, but this is producing a lot more tension than a single piece of paper. And so the way that we change how much tension is produced so that we can lift something heavier or be gentler and lift something lighter. And this, it all goes back to the number of cross bridges, right? The more cross bridges you have, the more tension, the more strength there is in the muscle. Okay? So as long as cross bridges can form, and more and more means more and more tension can be produced. Furthermore, we know that every single cross bridge cycle requires a single ATP in order to fuel it. And so as long as you have enough energy available for the cross bridge cycle, you can produce as much tension as you have cross bridges. Okay, so in this video and future videos, you are going to learn about 4 different factors that affect tension via the number of cross bridges that can form. The first that we'll talk about is the length of the fibers. So how long is the muscle cell when it begins the contraction? How quickly do you stimulate it? That is, how many action potentials by motor neurons do you have? The number of fibers you stimulate. So how many muscle cells within the entire muscle are stimulated? And finally, the type of muscle fiber. Not all muscle cells are created equal. So again, let's start by talking about the length of the fibers. What we can see here is a sarcomere. So Z disc, actin, Z disc, actin. The blue is myosin, here is the M line, and on myosin we can see the little myosin heads. In this scenario, before the muscle cell starts to contract, before it is stimulated, it is at such a length where there is significant overlap between the myosin and the actin. So as soon as the muscle cell is stimulated, all of these myosin heads can form cross bridges with actin and pull as much as they possibly can. Okay, so very much a lot of opportunities to form cross bridges. Okay, we can also see that there's a lot of space for the actin and myosin, or essentially for the Z disc to shorten before it hits the myosin. Okay, so with my hand scenario here, we can see that my fingers are overlapping, so cross bridges can form, and as soon as the muscle cell is stimulated, there's a lot of space to go to bring these Z discs together to shorten that muscle cell. Okay. If the muscle cell is already shortened passively before the muscle cell is stimulated by the motor neuron, yes, there are a lot of opportunities for cross bridges to form. Right? So we can see a pretty significant zone of overlap here. Myosin and actin can interact in many different ways. Therefore, lots of cross bridges equal lots of strength, lots of tension produced. The problem with the muscle already being shortened before it's stimulated is that there's really nowhere to go. Right? There's nowhere for this muscle to actually shorten, or the sarcomere to actually shorten to. So in my hand example here, here we have lots of zone of overlap. So my fingers are touching each other, lots of cross bridges can be formed here, but when the muscle is stimulated, it's already contracted as much as it can possibly go. It cannot contract anymore. So this is the shortest length of the sarcomere that we have. No matter how many cross bridges we have, we can't physically shorten anymore. Third example. On the other end of the spectrum, here we have, in this case, no zone of overlap. Right, so no zone of overlap or very short zone of overlap if the muscle is already stretched before you trigger it to contract. There are not many or any opportunities for cross bridges to form. As a result, even if the muscle is stimulated, you can't form cross bridges. No cross bridges or fewer cross bridges equal less tension. Right? Less tension, less strength can be generated. Okay, so in this example, there's very little zone of overlap or no zone of overlap. And so when active sites are uncovered, cross bridges can't form, and therefore you can't physically shorten the muscle cell or the sarcomere and ultimately the muscle cell. So ideally, you need kind of an intermediate length. So there's a zone of overlap, so cross bridges can form, but also you need some space for the sliding filaments to actually slide. Okay, and so we can experience this. Think about stretching a muscle or shortening a muscle and how much strength you can generate with it. So the example I'm going to use is my forearm muscles right here. These muscles, their job when they shorten is to pull the fingers and pull the wrist down into a fist. Okay? So this is shortened and this is longer of these muscles right here. Okay, so when you make a fist here, right, you can make a pretty strong fist, right? And if you make the fist, you might be able to feel the muscles moving here. So they're contracting, they are shortening, all those sarcomeres getting shorter. Okay? If you shorten the muscle before you contract it, before you stimulate it, that is, here, the muscle cells are actually shorter than they were before. Okay, so when you start the contraction, there's really nowhere for the actin and myosin to go, right? The Z discs are already right up against that myosin. And so when you try to make a fist here, right, with the muscle fibers already shortened, it's kind of pathetic, right? Like you really can't make a fist and it's really uncomfortable and feels super weird, right? So when the muscle is shortened already, you're not generating much tension. On the other hand, if you lengthen these muscles before the contraction, so back here, okay, here the muscles go up and over, and so they're much longer than they normally are. So if you try to make a fist here, again, it's really hard. Right? Not very much tension is able to be generated because there's very little zone of overlap in an extra stretched muscle. Okay, so let's look at a graph of this. Okay, so this is called the length tension relationship. We can see those images that we looked at on the previous slide. On the x-axis here is the length of the muscle at the beginning of the contraction. And here's the tension, right? So how much of the maximum tension of this muscle can be generated when the muscle starts out really short or when it starts out really stretched. And so we can see there is an optimal range where the zone of overlap is adequate to generate a lot of force, but there's still plenty of space for the shortening of the sarcomeres. We can see that as you stretch the muscle farther, right, farther and farther stretched, the less tension you can generate. Similarly, if you shorten the sarcomeres, right, you also can generate less and less tension. Okay. As we can see down here, the optimal resting length to generate the most tension in a muscle contraction is 75 to 130% of the optimal length. So actually, a little bit stretched is good, just a teeny bit, to generate the most tension. Okay. Up next, you'll learn about another way that we can affect the tension generated by a muscle.